Welcome back, everybody, for another deep dive. This time we are going to be talking about how protein interaction networks and RNA sequencing data can be used to identify promising therapeutic targets.
Sounds pretty complex.
Well, maybe, but hopefully by the end of this deep dive, we can make it feel a little bit more approachable.
I like that.
So to get started, I guess the big picture here is that we're basically talking about having like a blueprint of the cell's machinery and then being able to see in real time what's running too hot or maybe what's not running at all.
Yeah, that's a great analogy. By combining these two approaches, the protein interaction networks, which we often shorten to PPI networks, and the RNA sequencing data, we can get a much clearer picture of what's going wrong in a disease, and I think even more importantly, where we can intervene, to actually make a difference.
So let's start with those PPI networks. They're basically like a map of how all the proteins within a cell interact with each other, right? But why is that so important for drug discovery?
Well, pretty much everything that happens in a cell involves proteins interacting with each other in some way.
Makes sense.
And these interactions form these really, really complex networks, kind of like a giant social network, but for proteins. And within these networks, certain proteins act as what we call hubs.
Hubs?
Yeah, so think of them kind of like major airports, lots of connections, lots of traffic coming and going.
Okay, I think I see where this is going. If one of these hub proteins has a problem, it can cause a ripple effect through the whole system.
Exactly. It's like when there's a delay at a hub airport and suddenly flights are messed up all over the country. And because these hub proteins are so crucial, they often end up being prime targets for new drugs. In fact, a 2022 study by Torrielli and his colleagues found that focusing on these PPIs has become a major trend in drug discovery.
Well, that makes a lot of sense. Many diseases are essentially glitches in these protein interactions, right? So where does RNA sequencing fit into this whole picture?
So RNA sequencing, or RNA-seq as it's often called, is like checking the volume levels on all those protein interactions.
Volume low.
Yeah, it tells us how much of a specific protein is being produced by a cell.
So it's kind of like seeing which genes are turned up really loud and which ones are barely whispering.
Precisely. Think of it as a volume knob for protein production. And by combining RNA-seq data with the PPI network, we can see which of those highly connected hub proteins are also being overproduced in a disease. It's like seeing which of those busy airports are also having major traffic jams.
Okay, that makes sense. So it's not just about finding proteins that are interacting, but also seeing which ones are really cranked up and potentially causing problems. It's like adding a whole new layer of information to that map.
Exactly. And this combined approach has some big advantages.
Like what? Give me the highlights.
Well, for one, it increases our specificity. We're not just looking for any overexpressed protein, but for those that are also hub proteins. So it helps us filter out the noise and focus on the most promising targets.
So it's like using a really fine-tuned filter to zero in on the most important players.
Precisely. Plus, this approach can help us uncover totally new insights into how diseases work.
Ooh, I love those aha moments. Do you have a good example of that?
Definitely. Back in 2025, On and their team used this combined approach to discover a cluster of histone proteins that were specifically linked to a particular type of breast cancer.
Histone proteins, those are the ones involved in packaging DNA, right?
That's right. So essentially, they found a hidden control panel for this specific type of cancer, and they were able to do that by looking at both the protein interactions and the RNA expression levels.
That's amazing. It really highlights how powerful this approach can be and how it can help us find those hidden connections that we might have missed otherwise. But I imagine it's not always smooth sailing, right? There must be some challenges with this approach too.
You're right, there are. One of the biggest challenges is variability. Both PPI networks and RNA-seq data can differ depending on how they were generated, the samples that were used, even the analysis methods. So it's not always a perfect apples-to-apples comparison.
So how do scientists deal with that variability? Doesn't it make it more difficult to interpret the results?
It definitely adds a layer of complexity. Researchers have to be really careful about how they collect and analyze the data, and they need to consider all the potential sources of variation. Another important thing to remember is that just because a protein is overexpressed, it doesn't automatically mean it's a good drug target.
So it's not as simple as find it and target it. What else do scientists need to think about?
Well, some proteins might be structurally very difficult to target with a drug. Even if they are playing a key role in the disease, it's like finding a lock but not having the right key. That lock is crucial, but you might not have the tools to open it.
That's a great analogy. So while there are definitely challenges, there have also been some exciting success stories using this combined approach. What are some of the highlights?
Well, for example, in 2024, Trepti and their team used artificial intelligence to map and score protein interactions to try to find potential inhibitors of SARS-CoV-2 proteins. Essentially, they were trying to block the virus from replicating.
So they used AI to help them sort through all that data and find the most promising leads. That's incredible. What did they find?
They actually discovered a completely new way to block virus replication by targeting a protein that no one had really considered before.
Wow. So it's not just about finding new drugs, but also finding new ways to use existing drugs or even repurposing drugs that were originally designed for something else. That's really impressive. Were there any other success stories that really stood out to you?
Absolutely. There's the work by Carraro and their team from 2022. They showed just how powerful these multi-omic approaches can be. They integrated transcriptomic data, which is essentially RNA-seq, with chromatin state data to get even deeper insights.
Okay, hold on a second. Chromatin state data. That sounds like we're getting pretty deep into the weeds here. Can you break that down for us non-scientists?
Sure. So imagine DNA as a really, really long thread that's wrapped around these spools.
Okay, I'm picturing it.
Chromatin data tells us how tightly that thread is wound, which affects which genes are turned on or off. By combining this information with RNA-seq, data researchers can see not only which genes are being expressed, but also how their activity is being regulated.
Ah, so it's like getting a behind-the-scenes look at the whole production process, not just seeing the final product. That's fascinating.
Yeah.
And this led them to new therapeutic targets.
Exactly. By understanding how gene expression is being controlled, they were able to identify new targets that could be exploited with drugs.
That's incredible. It seems like we've gone from basic maps and traffic reports to having this amazingly detailed blueprint of the cell. But what really blows my mind is the role of AI in all of this. It seems like it's completely transforming this whole field.
It absolutely is. The amount of data that's generated by PPI networks and RNA sequencing is enormous, and traditional methods of analysis just can't keep up. AI and machine learning are essential for making sense of all this information and identifying patterns that humans would likely miss.
So it's like having a superpowered research assistant that can analyze mountains of data and point out the most interesting findings.
That's a great way to think about it. And AI can do so much more than just basic analysis.
Like what? What other tricks does it have up its sleeve?
For instance, AI can be used to predict how a particular drug will affect an individual's unique PPI network.
Wait, so we could potentially tailor treatments to a person's specific protein interactions. That's personalized medicine on a whole new level.
Exactly. And AI can also streamline the whole drug discovery process, especially in high-throughput screening, where you're testing thousands of potential drug candidates. Instead of taking years to analyze all that data, AI can help us find those needles in the haystack much faster.
So it's like having a fast-forward button for drug discovery. This is really revolutionary stuff. But before we get too lost in the world of AI, let's take a step back and recap what we've learned so far.
Sure. So the main takeaway is that PPI networks and RNA sequencing are incredibly powerful tools on their own, but when you combine them, they become a real force to be reckoned with in drug discovery. They allow us to understand disease mechanisms in much greater detail and help us zero in on the most promising targets for new therapies.
It's like having a multi-pronged attack on disease, increasing our chances of success. But even with all these advances, I'm guessing there's still a lot we don't know, right?
Of course, science is all about asking questions and pushing the boundaries of knowledge. The more we learn about PPI networks and RNA sequencing, the more new questions will emerge and new challenges will arise.
That's what makes it so exciting. There's always something new to discover. But on that note, we'll take a short break and come back to explore some of the most intriguing possibilities this research opens up, especially when it comes to personalized medicine. Stay tuned. Welcome back. We've been talking about how this combination of PPI networks and RNA sequencing is giving us a much deeper understanding of disease and leading to some incredible breakthroughs in drug development. But it seems like this research has even broader implications.
Absolutely. We're talking about potential impacts on public health initiatives and even the economy.
Okay, so let's start with public health. How could this research actually change things on that front?
Well, just imagine being able to identify people who are at high risk for certain diseases based on their unique genetic and protein profiles.
That would be incredible. We could actually be proactive instead of just reacting to health problems after they occur.
Exactly. We could implement early intervention strategies, maybe even encourage lifestyle changes that could dramatically reduce the incidence of those diseases. It would be a game changer for healthcare systems around the world.
And from an economic standpoint, I imagine this kind of research is going to lead to a ton of innovation.
Oh, absolutely. We're already seeing a surge in investment in biotech companies that are focusing on this kind of research, and that's creating new jobs and giving a big boost to the economy. It's a win-win.
It sounds like this research has the potential to really change the world for the better. But with any new technology or approach, there are always those potential downsides and risks to consider.
Right, you always have to be mindful of the potential downsides. One concern is that as we delve deeper into this incredibly complex world of protein interactions and gene regulation, we might unintentionally disrupt some very delicate balances within the body.
So it's kind of like the butterfly effect.
Yeah.
One small change could have unforeseen consequences.
Yeah, that's a good way to put it. We could end up triggering unexpected side effects or maybe even creating new health problems. It's a reminder that we need to proceed with caution and humility as we venture into this uncharted territory.
Makes sense. It sounds like this research has the power to do a lot of good, but it also comes with a lot of responsibility.
Absolutely. The more we understand about the intricacies of our biology, the better equipped we'll be to make informed decisions about our health and well-being, both as individuals and as a society.
Well said. It's been incredibly insightful to hear about all of this.
Yeah.
But before we wrap things up, I'm curious, are there any suggestions you have for our listeners who want to explore this topic further on their own?
There's a wealth of information available online and in libraries. Don't hesitate to delve into the research we've discussed and really explore these fascinating discoveries.
Sounds like a great place to start. And with that, I think it's time to shift gears and delve into some of the more ethical considerations surrounding this research. Seems like there's a lot to unpack there. Welcome back to our deep dive into the world of protein interactions and RNA sequencing. It's been a fascinating journey so far. We've talked about the science, the breakthroughs, and even the economic implications.
Yeah, it's amazing to see how this research is rippling outwards, touching so many different aspects of our lives.
It really is. But like with any powerful technology, there are always those ethical considerations that we can't ignore.
Absolutely. Ethics are always a key part of the conversation, especially as this research continues to advance.
So let's talk about some of those potential downsides or risks. What are some of the things we need to be mindful of as we move forward?
Well, one big one that comes up a lot is the potential for genetic discrimination. If employers or insurance companies were to get access to somebody's protein interaction and gene expression data, could they use that information against them?
Yeah, that's a really scary thought. It's like the whole pre-existing condition debate, but on a whole new level.
Exactly. It raises all sorts of questions about privacy and fairness. Like if someone is deemed high risk based on their genetic profile, will they be denied coverage or face higher premiums?
It seems like we'd need some really strong legal protections in place to prevent that kind of discrimination.
Absolutely. We need to make sure that this kind of information is used responsibly and ethically for the benefit of individuals, not to penalize them.
Okay, so genetic discrimination is a big one. What other ethical concerns are on your radar?
Another big one is access and affordability. We've touched on this before, but these cutting-edge therapies, they have the potential to be incredibly expensive.
Yeah, and we don't want to create a situation where only the wealthy can afford these life-changing treatments.
Exactly. We need to be really careful about not exacerbating existing healthcare disparities. Everyone deserves access to these advancements, not just the privileged few. It's a matter of social justice.
I couldn't agree more. Equal access to healthcare should be a fundamental right, not a luxury.
Right. And then there's the issue of informed consent. Do people really understand the implications of sharing their genetic and protein data? Do they know how that information might be used down the line?
That's a really important point. We need to make sure that people are making informed decisions about their healthcare and that they're not just blindly handing over their data without understanding the potential risks and benefits.
Transparency is key. And it's not just about having people sign a consent form. It's about making sure they actually understand what they're agreeing to.
It's about empowering patients to be active participants in their own healthcare decisions.
Right. And honestly, I think that comes down to better communication. Scientists and doctors need to get better at explaining complex scientific concepts in a way that's clear and understandable to everyone.
So it's about bridging that gap between the scientific community and the public.
Exactly. It's about making this information accessible to everyone, regardless of their scientific background.
Well said. It sounds like there are a lot of important ethical considerations to keep in mind as this research continues to evolve. But overall, wouldn't you say that the potential benefits of understanding protein interactions and gene expression far outweigh the risks?
I truly believe they do. The
Hmm.
insights we're gaining are groundbreaking and have the potential to revolutionize medicine as we know it.
It's an exciting time to be alive, that's for sure.
Okay.
But before we wrap up this deep dive, do you have any final thoughts or words of wisdom for our listeners?
Absolutely. If this conversation sparked your curiosity even a little bit, I encourage you to keep exploring. There's a ton of amazing research out there that's just waiting to be discovered. Don't be afraid to dive in and learn more about this incredible field.
That's great advice.
Yeah.
Knowledge is power, right? Well, on that note, it's time to wrap up this deep dive into the world of protein interactions and RNA sequencing. It's been an absolute pleasure having you with us today.
Thanks for having me. It's been fun.
And to all our listeners out there, thank you so much for tuning in. Until next time, stay curious and keep exploring the wonders of science.